So a really great feature of our symposia is the chance to showcase and discuss the work done by our trainees. So our first trainee speaker will be Elyn Rowe, who's completing her PhD degree at the CBR, who will be speaking about blood products from diabetic donors. So Elyn, thank you very much.
Okay, yeah, thank you very much for the introduction. Once again, my name is Elyn Rowe. I am a PhD candidate at the UBC Center for Blood Research, formerly supervised by Dr. Dana Devine and now supervised by Dr. Jim Johnson. And today, I'm really going to take you through part of my journey, at least, to determine what's in the bag of blood products from donors with diabetes. So we talked a lot this morning about, in the field of transfusion medicine, the need to meet demand, and that's a huge problem. But if we can tackle that, then the next really interesting question, I think, is to really understand what's in the bag of each unique blood product, as they're not all equal. So because blood products come from a wide range of human donors, they have inherent biological variability in their characteristics, which seem to be linked to the donor. And now that may initially sound like a bit of a concern, but it's actually something that we could leverage if we could understand it better. So take conditions that require chronic transfusions as an example. So in this case, we would want products that are long-lasting and have lower clearance to reduce the number of transfusions required for the patient. And as you'll see in other talks later today and in some posters, this may look like specific characteristics of the blood products, such as increased deformability of the red cells. So really what I'm saying is if we could identify sources of this variability, this could lead to tailored applications potentially of blood products to improve patient outcomes. So now we pivot to diabetes, which is really an umbrella term for conditions that result in sustained high blood sugar levels or hyperglycemia, which can damage cells and tissues in the body. And so it then may follow, and there actually is some evidence, that blood components are altered in people with diabetes, perhaps due to this hyperglycemia. So type 1 and type 2 diabetes have different etiologies and treatment approaches, and importantly for this talk, they have different policies around the world governing eligibility of these individuals to donate blood. So in Canada, most individuals with either type of diabetes are eligible to donate blood, but in most blood centers worldwide, all donors with type 1 diabetes are deferred. And now, importantly, these heterogeneous eligibility policies are not rooted in concrete data about the quality of the products, and there's currently actually no data on the quality of products from donors with type 1 diabetes. And further, as global diabetes prevalence, mainly of type 2 diabetes, as you can see here, is rising, these donors will make up a larger fraction of our donor pool. So it's something that we really need to understand. And this really feeds into my key research question here of does diabetes drive differences in blood product characteristics or quality over storage? So as glycemic control is not actually assessed in routine donor screening, we started by screening a sample of active Canadian Blood Services donors. And so what we did here was we flagged donor records such that we would receive tubes of blood following routine donations across Canada so that we could measure glycated hemoglobin, or HbA1c. And this is a marker of average glycemic control. So you can imagine if we have high blood sugar levels, there's more sugars available to glycate hemoglobin, HbA1c will rise. And so as there's typically this healthy donor effect in our populations of blood donors, we thought that perhaps all of our donors with diabetes would be really well controlled, and we might not have to maybe worry as much, but this was actually not the case. So as we can see here, several donors, especially males over here, have very poor glycemic control indicated by high HbA1c, and actually that the average HbA1c in both groups is above the target set by Diabetes Canada of 7%. And so really what we found here was that nearly half of Canadian Blood Services donors with diabetes have suboptimal glycemic control, which may then feed into potentially marked differences in the blood products. So then we followed up looking into exactly that. So in the next phase of this study, we then recruited donors with each type of diabetes and age and sex matched controls to donate a whole unit of blood for research. So here is a demographics and clinical test table of these donors, and as shown by the p-value columns here, these groups are fairly well matched apart from HbA1c as expected. And importantly to note, the average HbA1c in our donors with diabetes here is lower than the average that we saw across the CBS donor population. And this sample actually has relatively good glycemic control. It's below that Diabetes Canada target of 7. And we have similar HbA1cs between our diabetes groups. And so then from these whole blood donations, we then made leukoreduced red cell concentrate and single donor buffy coat concentrate, platelet concentrate, and characterized these products across their routine storage periods that might be used in hospitals with a battery of different assays. So starting with some key quality parameters used by Canadian Blood Services over storage, we have platelet pH and red cell hemolysis across the storage periods on the x-axis here. And while type 1 diabetes products from donors with type 1 diabetes in blue on the left panels are comparable to their matched controls, we actually do see some marked differences in the type 2 diabetes group in red here, where we have they're significantly different with lower pH and higher hemolysis by the end of storage. And this really indicates that the quality of these products from these donors is declining faster than their age and sex match controls. And while these differences are not approaching the standard CBS quality cutoffs that are accepted for premature expiry of products, we do actually, interestingly, also see statistically significant functional decline in both platelet responsiveness to a weak agonist and in red cell deformability in response to osmotic stress by the end of storage, again, only in donors with type 2 diabetes. However, if we then return to this global context I was talking about of pinpointing sources of variability, we do see pretty wide 95% confidence intervals indicated by the shaded parts on the graphs here, indicating that not all variability in these parameters is driven by diabetes alone. There's likely a whole bunch of different factors that we haven't even touched on here. So really, what can we make of all of this? First, we found that nearly half of Canadian blood donors with diabetes have suboptimal glycemic control, which was a bit surprising, but this is actually in line with the broader Canadian population of individuals living with diabetes, so it's not like we have a particularly unhealthy group of people with diabetes donating blood. And then in our deeper characterization of the products, we found that platelet and red cell concentrates only from donors with type 2 diabetes have statistically significantly steeper decline in quality relative to controls, but these effect sizes may not be clinically significant. They weren't nearing those CBS quality cutoffs. We also found, interestingly, that type 1 diabetes does not appear to have an impact on platelet or red cell characteristics or storage performance. And this is really important information for other blood services worldwide who are considering expanding their donor eligibility criteria to include these donors. And also, as glycemic control was relatively even between our diabetes groups, and we saw different trends but in type 1 versus type 2 diabetes, these differences that we saw in our products from donors with type 2 diabetes is likely not driven by glycemic control alone, and we may need to look to other factors to explain this. So as for next steps, we have sent specimens off from all of these products for metabolomics analysis, and we hope to gain some insight into mechanistic differences in storage from that, and I'm very excited to see where this data takes us. So I'd like to thank my supervisory committee and everyone who's helped along the way. Thank you.
Thank you very much. Very nice talk.
Thanks.
I was wondering, what do you hypothesize? Where does the difference between type 1 and type 2 diabetes come from?
Yeah, so there was actually just recently a paper published indicating that metformin might be a causative agent for driving the decline in quality of at least platelets over storage, and that is due to the mechanism of changing the metabolism essentially in the platelets. Now there's no data on why we're seeing higher hemolysis in the red cells, but it could be something to do with medications taken. In the quick look that I had, we had some of my donors were on metformin, some were not. It doesn't seem to be that that's the main driving factor in this. Our donors not on metformin, we do still see that decline in storage quality. It could be dyslipidemia, which we see more in donors with type 2 diabetes compared to type 1. It could be, we have BMI differences in our donors with, there are all these other factors that come with type 2 diabetes that make those donors a bit more different than their age and sex match controls. So I'll be in data analysis graph land, I'm sure, trying to figure that out before my defense.
Thank you.
Thanks.
Are you quantifying the amount of recombinant insulin in the samples? Type 1, they get all the time insulin, right?
Yeah,
is that a problem for autoimmunity potentially down the road?
Like post-transfusion, like if the products contain recombinant insulin and then are then transfused. We're not quantifying that here, but that is something interesting to think about. Yeah.
That was a great talk.
Thank you.
Thank you very much for doing that and your work. I'm just wondering, do glycosylated hemoglobin red cells, are they prone to hemolysis more than are non-A1C?
Not that some collaborators at the University of Alberta have found so far. And because our HbA1c levels were similar between diabetes groups and we're only seeing that uptick in hemolysis from type 2, it seems that doesn't seem to be a driving factor. And when I've looked at correlations, HbA1c doesn't seem to be correlated with hemolysis levels, but...
Can those cells be purified?
Ones with higher levels of HbA1c? I have not tested it myself, but I mean,
they must have a unique carbohydrate structure on their surface, right? There's lots of antibodies to carbohydrate antigens.
Yeah. Yeah, I'm sure surface proteins are also increasingly glycosylated, and that may correspond with
it might be a subpopulation that's causing your storage problem for hemolysis.
Yeah, it could be. But yeah, and actually in this sample we had higher average HbA1c in our type 1s, and we're not seeing that increased storage hemolysis. It kind of leads me to believe that may not be the case, but it could be an interesting line to follow up on for sure.
Are there any other questions? No? Thank you very much.
Thank you.